Again, that's very topical, myrtle rust, susceptibility of Manuka and other Myrtaceae species associated with honey production to myrtle rust. Beccy Ganely, thank you.
Kia ora tātou, ko Beccy Ganely toko ingoa, ki mahi ana ahau, ki te rangaha ahu marakai, ki te puki. So I'm Beccy Ganely, I'm from Plant and Food Research and I'm the Science Group Leader of the Plant Pathology Group. And as was said, I'm going to be talking today about the susceptibility of Manuka and some of our other Myrtaceae to myrtle rust. So this work's been done by a huge team of scientists, all listed up there, from New Zealand, Australia and South Africa. So first off, what is myrtle rust? Hopefully everyone in the room knows what myrtle rust is, but as Samuel said, need a bit of background just to make sure that everyone's sort of up to date with exactly what the disease issue is and what sort of the state of play. So it's caused by the pathogen Ostrobaccinia acidii. It's a biotrophic pathogen, which means it feeds on live tissue. Worldwide, we know that there are lots and lots of different strains of myrtle rust, and what's present in New Zealand is known as the pandemic biotype, and it's also the same strains that are present in Australia. It has a really, really wide host range, over 400 Myrtaceae hosts, and this is really unusual for a rust pathogen because usually they only have one or two hosts. So having one that has over 400 is really, really amazing or unusual. We've got lots of different exotic and native Myrtaceae in New Zealand, and part of the question is what's the impact of this pathogen on those Myrtaceae species? So the pathogen was first found in the mainland in May 2017, and I've got a picture up here, I'm hoping this is what this is, of where it's been found so far. So the yellow ones are positive detections, there's 3 red ones which are some new recent detections, and the blues are areas that were sampled and the rust wasn't found at the point in time of when it was sampled. Not meaning to say that it's not present there now, but at that point in time it wasn't present. So why are we worried about this disease? Well, the problem that it can cause can range hugely. Some plants are completely resistant or tolerant to the pathogen, but then it goes all the way up to the extreme, which is what they've seen in Australia, where you can have susceptible species which after repeated infection end up dying, and then what they're seeing as a consequence 5 years after they first detected the pathogen in Australia, they saw the first localised extinction of a couple of their most susceptible species, which are the native guava and the malletwood. And that's just 2 of the species that they know have had localised extinctions. There's potentially more. And then in 2018, they've realised that they're actually looking at landscape level extinction of those species. So that's the worst case scenario, and that's what we're the most worried about with what could myrtle rust do in New Zealand. And then part of the consequence of that as well is what replaces those Myrtaceae in an ecosystem when they go. So in New Zealand, when we first detected myrtle rust, MPI put an incursion response through, and as a result of that, through the maps that you saw previously, huge amount of surveillance was done. So this is old data, but it's no longer kept up to date because it's moved into long-term management for myrtle rust. But clearly you can see that they had surveyed a huge number of plant species. And you can also see which ones are clearly susceptible, and ones that aren't on this list either may be resistant or tolerant or otherwise just might not have been exposed to the spores. So what we've been concerned about as a scientist is that when you look at a list like this and you see 20,000 Manuka plants and only 3 that are infected, what does that mean? It just means that we found 3 plants that were infected, and that's about as much of the information you can take from that. We don't know how susceptible they are. We don't know if the remainder of those plants that were looked at had never been exposed to the spores, and is that why they weren't infected, or is it because they're resistant or tolerant? So that leads on to the next one. That's actually the one plant that was infected, and there's the one lesion. One of the first plant that was found, that was Manuka, sorry. So that leads on to what we did, which was looking at how susceptible is Manuka and some of New Zealand's other Myrtaceae to myrtle rust. So we collected almost 1,300 families of Manuka across New Zealand, collected the seed from it, sent it over to Australia, where it was grown and tested against the pandemic biotype, so the same strain that we have present in New Zealand. But it was done in Australia because they were set up to take large groups or batches of material to test, and also at that point in time we were unable to do any of the testing in New Zealand. And that's a picture of them, some of the seedlings that are getting screened. So what did we find? We found this, well, this graph here is looking at the resistance of the populations that were screened. And so at the bottom, we've got all the different families, and at the top, we have the percentage of plants within that family that were resistant. And so as you can see, there's a huge variation in resistance. What was also quite interesting, we found two types of resistance, leaf resistance and a stem resistance. So it went from families like this, where 60 to 70% of the family are resistant and they have both a stem and a leaf form of resistance, through to ones like this, when no members of the family had any form of resistance. And for where it's not resistant, the converse is susceptibility, there was a variation in how susceptible the plants were. So overall, we're really finding that there is a wide variety of resistance and susceptibility, but the presence of resistance of Manuka is a real positive, because that does mean that we have the potential for breeding programs to make sure that progenies going through are resistant to myrtle rust. One of the things that we did find, which was incredibly concerning, is the flowers of Manuka were able to be infected by the pandemic biotype, and that's clearly of interest, of concern for any of the Manuka-based industries in New Zealand, including honey production. As well as finding the variation in susceptibility of Manuka, we also found the same with some of the other Myrtaceae we tested, including Pahutakawa. But what the next steps are really is just to see how New Zealand Manuka is going to respond to myrtle rust in the New Zealand environment. So at the moment, the inoculum levels of this pathogen are actually quite low, but what will happen as those inoculum levels increase, are we going to see more Manuka being infected, and how will those resistance mechanisms come into play with the level of disease, and more importantly, how the flower's been infected. So I've talked about the pandemic type biotype, but that's only one of the strains of many that of myrtle rust that are present, and so we're really concerned about what would happen if one of the other strains of myrtle rust should come to New Zealand. They're easily spread, and so part of this work was also to test against the South African strain. This one's gone to South Africa, causes huge problems in some of the Myrtaceae that they have there, and so the question was, well, how susceptible or resistant will our Myrtaceae be to this strain? So we sent through Manuka, Kanuka, Pahutakawa and Manuka rawiri seed across to South Africa. It was grown and inoculated against this strain. In short, we found that all of the species were susceptible. Kanuka was definitely the most least susceptible, Manuka and Pahutakawa the most, and the Manuka rawiri was actually a really small sample size, so not really entirely sure. It was susceptible what we tested, but it was only a very small number of seed or plants that were tested. Again, which was really concerning, we're also seeing these flowers are getting infected with myrtle rust, and in this case the flower did die. And as well as you can see some of the lesions we have, and you can see sort of the variation in what you're seeing with the myrtle rust on the 4 different species. So this is just to give an example of some of the things we look at and that level of susceptibility with these different species. So we've got this one at the top here, the green is the Manuka. We looked at the red spots, which are often the first symptoms. We looked at the iridineospores, which are the yellow ones that we always talk about. But then also with this one, it has teleospores, and these are sexual. And so they're really concerning from a pathogen point of view, in that it causes sexual recombination, so you can end up with strains which are more virulent than the ones that you currently have. And also for stem infections. And as you can see, the Manuka was clearly the most susceptible of all, but positively, not everything's getting infected. So where it's not hitting up there means that you've got resistant or tolerant populations. So just in summary of what I've gone through, for the pandemic biotype, There is definitely susceptibility and resistance, and as I said, this is really positive and from a breeding perspective. Yes, we're probably going to have some plants out there that are going to die. We don't, or not die, but get infected, sorry, I should say, and the material on them may die. But then also there should be plants out there that are either fully resistant or have a really high level of tolerance. Clearly, the infection of flowers is a major concern. And then for my message for everybody in the audience, if you're going out there, is to be really vigilant about when you're in a Manuka stand. Does it have myrtle rust? And if it does, can you remove those plants or parts of the plants to try and keep the inoculum level low, and also preventing further spread of it. We really need to keep an eye on what's going to happen with Manuka as the inoculum levels increase. And what we also don't know is what is the impact of insects. For instance, will the bees forage on spores instead of the pollen? And one of my most great ones is conservation of germplasm, especially for a breeding program. Finally, for the overseas strains, again, susceptibility and resistance. Infection of flowers, again, is a major concern, and that's awareness to make sure we're continuously vigilant of making sure that these new strains aren't accidentally brought into New Zealand on people who are moving infected plant material, or if you've gone somewhere that overseas that has myrtle rust, make sure you clean your clothes, your shoes and your equipment, and don't bring it back into New Zealand to try and halt the spread. And then my final slide is just to thank Mana Whenua in particular, who've allowed us access to the land and the plant material and to send it overseas for testing, and Rebecca Campbell for the surveillance map. And then these are all the different groups that have been involved, whether they funded or contributed to the research. So thank you very much.